Hi everyone. Thank you so much for attending our first Neurodegeneration conference for early career researchers. It looks like it's going to be a spectacular event and I'm really excited to get started. So my name is Rebecca Northeast and I am the technical specialist for Europe at Protein Tech. And I'm just going to take you through an introduction and a brief run through of the day. So at Protein Tech, we decided to host this meeting because we wanted to provide a platform for early career researchers to showcase their work at a time when attendance at physical meetings is sadly not possible. We are so pleased to be able to bring together an international audience and enable the exchange of ideas in the absence of SFN. Here at Protein Tech, we love attending SFN and getting to interact with all of you. And so this week, we are hosting our own Neuro Week, centering on this meeting and taking our interactions virtual. We are offering free samples of our 10 most popular neuroantibodies, as well as our very popular iHeartBrains tote bag. This is the schedule for today. We will start with an introduction from our session chair, Dr. Maina, followed by 4 short talks from the ECR speakers. After this, there will be a questions and answer session. Following the question session, we'll then have our keynote lecture from Professor Tara Spires-Jones from the University of Edinburgh, followed by questions for Tara. During both the question sessions, Dr. Maina will read out any questions submitted by the audience. So please feel free to type any questions into the chat function. We really want to encourage communication and collaboration. So if you could leave your email address alongside your questions, then the speakers will be able to reach out to you if your question is not answered. Alternatively, if you want to contact them directly, if you have any further questions, then their contact details will be on show during the question session. So now I'd like to introduce Dr. Maina. Dr. Maina is a research fellow at the University of Surrey in the UK. His research focuses on understanding the basic mechanism of disease in neurodegenerative diseases, with a focus on Alzheimer's disease. Dr. Maina is also a huge advocate for promoting science, especially neuroscience. Since 2013, he has founded multiple organizations focusing on enhancing the public understanding of science, especially in Africa. He has published articles about neuroscience in Africa and organized workshops to train African neuroscientists. He is also a co-organizer of the Society of Neuroscientists of Africa in the webinar series. So now over to Dr. Mahmoud to introduce the ECR speakers.
Thank you very much, Rebecca, for the introduction. And I feel very privileged to be invited to chair this meeting. And I'm also very excited and looking forward to all the fantastic talks. And based on the inbox that I've seen that some of the attendees are from, I think, India, and I think there are other people from all around the world. So it's really, really exciting to see that we've got this broad range of attendees from all around the world. I think you've done kind of a fantastic introduction about the nature of the meeting, but I would like to just emphasize on the fact that we've got only about 15 minutes for Q&A. So what I would encourage us to do is to send in our questions through the Q&A inbox session, inbox box on Zoom, and then let's try to be specific about who we want to ask the questions to. And apart from that, I will try to encourage us to probably drop our Twitter handles, because one of the key reasons of the meeting, based on what Rebecca has mentioned, is to create this networking opportunity between ECR. So if you could drop your Twitter handle, perhaps other attendees can follow you back and the conversation can continue on Twitter. Without really wasting time, I think I'm gonna go straight into introducing our first speaker, who is Henriette Haukedal. And she'll be talking to us about patient iPSC-derived neurons for disease modeling of familial Alzheimer's disease with mutations in presenilin 1. But without really giving her the stage, let me give a brief bio about her. So Henriette completed a bachelor's degree in biology at the Norwegian University of Science and Technology in 2015, where she specialized within cell and molecular biology. She followed that with a master's in human biology from the University of Copenhagen in 2018, where she worked on stem cell modeling of neurodegenerative disorders. And since 2019, she has been pursuing a PhD in neuroscience, continuing from her own MSc work at the Faculty of Health and Medical Sciences of the University of Copenhagen. So I think I've said a lot. Let me just give the stage now to Henriette to give us a talk. Henriette, are you with us? And can you share your screen?
Yes, working on it.
Fantastic.
Let's see. Perfect. I hope you can all see it now. Perfect. Then I'll start with my talk. So thanks for the introduction. So I'm a PhD student at the University of Copenhagen, and today I will present my one of my PhD projects, which is patient iPSC-derived neurons for disease modeling of familial Alzheimer's disease with mutations in presenilin-1. So I'll start with a brief introduction to Alzheimer's disease. So dementia affects more than 50 million people worldwide, and about 60 to 70% of all of these cases are caused by Alzheimer's disease, or AD, clinically presented with a progressive decline in memory. And this is an age-related disorder, making it an increasing health challenge today, and there's no currently available cure. So research within the area is crucial to get a better understanding of the disease pathology. And Alzheimer's disease can be categorized as either sporadic with no apparent heritability, or as familial, where you have specific mutations in one of three genes, either APP, presenilin-1, or presenilin-2, that is the cause of the disease. And in a neuropathological aspect, Alzheimer's disease is categorized by the formation of extracellular amyloid beta plaques in the brain and intracellular neurofibrillary tangles caused by tau phosphorylation. And together with these two hallmark, neuroinflammation is also seen in the brain of these patients with the Alzheimer's disease, as also for a lot of other neurodegenerative disorders. With that, I would like to thank my group, my research group, the Freude Group at the University of Copenhagen, with my main supervisor, Christine Freude, of course, our funding as well, and of course, Protein Tech for organizing and inviting me to speak. And my email address has been on every slide, so if you have some questions that's not been covered or we don't have time to cover later on, just shoot me an email anytime. Yes.
Fantastic, fantastic. Thank you very much, Henriette. It's such a fantastic presentation. And I hope that by next year we'll be able to hear you talk again about all the future experiments that you're planning to do. Okay, so I think without wasting much time, I would like to move on to the next talk. And the next talk is going to be given by Dr. Siobhan Crilly. And she'll be talking about chemical screening in zebrafish larval model of intracerebral hemorrhage for novel therapeutic discovery. But before giving you Dr. Siobhan, I think I should go on to tell you a little bit about what she's been doing before now. So Siobhan is a research associate at the University of Manchester in the Division of Neuroscience and Experimental Psychology. Her current research interests lie in the neurological disease modeling and finding therapeutic for brain hemorrhage. And Siobhan completed her PhD in 2019, for which she characterized why zebrafish larvae are excellent replacement model for brain hemorrhage, and continues to advocate the adoption of the model and reduction of animal usage in labs. And Siobhan, it's so great to have you today. Can you share your slide so that you can go ahead and start presenting your talk?
Yep.
Thank you.
Thanks for a great introduction, Mahmoud. So good afternoon from the UK. I'm Siobhan, and I'm from the University of Manchester. I'm going to tell you a bit today about how we model intracerebral hemorrhage in zebrafish larvae and have been using that model to conduct a chemical screen for novel therapeutics. So hemorrhagic stroke in the Western world is known as the less common and less researched type of stroke. It's accountable of 15% of strokes in population, but more than half of survivors suffer disability as a subsequent outcome of hemorrhagic stroke. So there's a huge global health burden that needs to be addressed. The physical tissue disruption of blood leaking into the brain causes mass effect and pressure, an increase in intracranial pressure in the brain, disrupting structures surrounding the hematoma, which contributes to cell damage. Downstream of where the blood erupts from the brain, erupts from the blood vessels, you get hypoxic tissues, which contributes to further loss of brain tissue, and toxic blood compounds, which aren't supposed to be in the brain, like heme, iron, and thrombin, build up around the hematoma. In the ongoing days, the hematoma contracts into this solid mass that really shouldn't be in the brain, and edema expands, contributing to further increase of intracranial pressure. And 10% of patients die immediately from this mass effect and increase in intracranial pressure. Secondary injury is contributed by neuroinflammatory response and the migration of peripheral cells into the brain parenchyma, and the increase of the blood-brain barrier breakdown and continued hematoma expansion. So around the world, intracerebral hemorrhage has a differing prevalence from the Western world. This is a graph to show you all of the countries in pink bars that have a lesser prevalence than the UK. The UK is down here, and the US is just next to it. And we have a prevalence of 30 per 100,000 people, which equates to about 20,000 cases in the UK. And that's why intracerebral hemorrhage is less researched than ischemic stroke, because it equates to about 15% of strokes. Whereas if we look at more severe countries, China has 2 million individual cases of intracerebral hemorrhage a year. And this is because of genetic differences and lifestyle differences. So despite the fact that intracerebral hemorrhage is a severe neurovascular emergency, there are no medical treatments for these patients, and it presents a global health burden. The only way that we can treat intracerebral hemorrhage patients when they present at hospital is to reverse anticoagulants in about 12 to 20% of patients who are taking them, reduce blood pressure to less than 150 millimeters of mercury, and in really special cases, in specific stroke care centers, we can do surgical removal of the hematoma to less than 15 mils. And this is an example here of the recent MISTI trial, in which a stent was inserted into the brain and TPA was used to dissolve the hematoma with better disability outcomes in 90 days. However, this isn't always possible in all countries in the world where intracerebral hemorrhage is a real global health burden. And we've recently reviewed the promising in vivo studies for repurposable drugs that have a real translational possibility for therapeutics for intracerebral hemorrhage. But currently, best medical practice is the best we can do for intracerebral hemorrhage patients. At Manchester, in the Kasha lab, we use zebrafish larvae as our model organism, which are small vertebrate, and they have many advantages over mammalian models. Firstly, they're really highly productive. So zebrafish pairing will lay about 200 to 1,000 eggs at once. They are very fast development, and at 3 days, you can see they have a fully developed central nervous system. They're see-through in larval stages, so we can non-invasively image internal structures. They're easily genetically modified, so we can generate transgenic lines and mutants for disease modeling. And in the UK, they are unprotected species up until 5 days post-fertilization. So experimentation is very quick and easy. In the Kasha lab, we have 2 models of intracerebral hemorrhage. Firstly, a drug-inducible model in which we treat the fish with atorvastatin at 24 hours, and a genetic mutant, which is a truncated version of beta-PICs, or GTPase. And by 3 days, we can non-invasively image the blood in the head to recreate an intracerebral hemorrhage. Now, this happens about 33 hours post-fertilization as a unique neurodevelopmental effect in the zebrafish. In regular development, endothelial cells will create tight junctions to form the blood-brain barrier. And this is aided by cholesterol rafts in the cell membrane for protein anchoring, and Rho GDPases like beta-PICs for actin cytoskeleton for protein anchoring. However, if we inhibit cholesterol using atorvastatin and knock out beta-PICs, then this results in leaky vasculature, and the blood can erupt from the blood vessel into the brain parenchyma. We use the transparent transgenic lines that we have in the University of Manchester to image the eruption of blood into the brain, to characterize the cell interaction and the pathological outcomes of intracerebral hemorrhage. And this was part of my PhD project I completed last year. So firstly, when we observe blood in the brain, we notice that this is associated with a severe region of dying cells in the hemorrhage individuals, which is characterized here by a transgenic line for cells. Additionally, they have a locomotor defect. So this is a startle response assay. A hemorrhaged fish for days after they exhibit hemorrhage have a locomotor deficit. And they also have an increased neuroinflammatory response, which we characterize by the increase of this MPEG-1 macrophage cells with more amoeboid morphology. And we have used live imaging to observe these macrophages phagocytosing their Nexin-5 positive cells. So in order to carry out our high throughput drug screen, we use the blood brain cell death as a consistent pathological outcome, as they have these clusters of brain cell death which are unique to the hemorrhage population and not observed in the non-hemorrhage siblings. And we also have a method of quantifying this overall total fluorescence. We use the spectrum library, which is 2,000 compounds, to screen for novel therapeutics in order to ameliorate the brain cell death. And we chose the apoptotic cells because we thought that these had more potential in order to rescue after a hemorrhage. We also decided to treat our fish after we'd already confirmed the presence of hemorrhage, rather than use a preventative strategy, as we believe that this had more translational approach. And we repeated our assay with positive outcomes and confirmed in our further locomotion assays as well. So out of the 2,000 compounds in the spectrum collection, 150 compounds reduced brain cell death clusters significantly. In order to cancel down this list a little bit more, we used the PREDICT and by random walk databases of drug-disease interaction based on drug-drug and disease-disease similarity in order to identify our compounds that were predicted to work in cardiovascular disease. And from this 150 compounds, we took on 6 further compounds for further study. We quantified the brain cell death that was visualized in these transgenic lines with treatment with 6 of these compounds, and found that there was significant reduction compared with vehicle, apart from 1. And in our preliminary study with all these compounds, we've also found trends to improving the functional deficit at 2 days after hemorrhage as well. Unfortunately, with compound C, the fish didn't survive to 5 days. So we're finding out that there are a little bit more to it than just reducing brain cell death. But drugs of note, A and B, which are both ACE inhibitors, and we found that they have significantly reduced the brain cell death associated with hemorrhage, and they have positive trends to restoring locomotor function at 5 days. Interestingly, ACE inhibitors are neuroprotective in other preclinical models of neurodegenerative disease, seen here an in vivo model of memory impairment in rats, and also in a glutamate model of toxicity in vitro. There have been clinical trials with ACE inhibitors in order to prevent the post-stroke disability in a pre-stroke kind of treatment strategy, and also angiotensin receptor blockers, which work on the same mechanism for ischemic stroke. Therefore, we took the INTERACT-2 dataset for ICH patients who'd been given blood pressure lowering drugs within 7 days of intracerebral hemorrhage to determine whether, if ACE inhibitors were delivered after an intracerebral hemorrhage, this resulted in better modified Rankin scores, which is a disability score at 90 days. And we found that in those patients, if they were given ACE inhibitors only after an intracerebral hemorrhage and not before and after, there was a significant improvement in the MRS scores at 90 days. With further sensitivity analysis, we found that these patients, if we excluded the patients that failed the swallow analysis before 7 days and were not able to receive treatment, we lost the sensitivity, we lost the significance in this result. So it goes to show that maybe ACE inhibitors, when delivered after a hemorrhage, need to be delivered as soon as possible. And there might be scope here for a clinical trial in which ACE inhibitors are not given orally and might be able to be delivered in IV at an earlier time point. Additionally, to show better disability outcomes in ischemic hemorrhage as well, ischemic stroke as well as in cerebral hemorrhage, implies that ACE inhibitors might be a suitable treatment to be given maybe in an ambulance, so patients can have treatment as early as possible. So our future questions in the lab are to determine the mechanism by which ACE inhibitors are exhibiting neuroprotection in our zebrafish, and whether these mechanisms are conserved in humans, and we can do that in post-mortem tissue. If we deliver ACE inhibitors early enough post-ICH, are they protective, and is this translatable to a clinical trial? So in the Brain Inflame group and the Kasha lab at the University of Manchester, we're using the fully translatable pipeline from zebrafish all the way to post-mortem patient tissue to look at the secondary injury responses to ICH, neuroinflammation and neurodegeneration, to see if we can find a treatment that can be applied widely and accessible for all integral patients across the globe. Thank you very much for listening. I'd like to thank the Kasha lab at the University of Manchester, all the collaborators for the paper that helped me acquire the data, the bioimaging core facility, the genomic technologies core facility, and the biological service unit, our collaborators at the University of Sheffield for sharing the spectrum collection, and at the George Institute and the Interact2 research group. Additionally, Protein Tech and Rebecca for organising the meeting today. Thank you very much.
Thank you very much, Siobhan. This is really a great talk, and I'm pretty sure a lot of people, especially those from Africa, will appreciate such model, because we've been advocating for use of small models like this back there. So thank you very much. And I think our next speaker is Dr. Cara Croft. Before going on to introduce her, can I invite her to start sharing her screen? So Cara will be talking to us about understanding the formation and clearance dynamics of neurofibrillary tau inclusions. And she completed her PhD at King's College London in 2016, where she worked with Wendy Noble and Dan Hunger. Following that, she started a postdoctoral fellowship with Todd Gold at the University of Florida and was awarded a Bright Focus Foundation Fellowship looking at the relationship between tau and neurodegeneration. In 2019, Cara was awarded a prestigious 5-year race against dementia fellowship working between the UCL and UK Dementia Research Institute at the University of Florida and the University of Florida to understand more about the genetics of Alzheimer's disease and Parkinson's disease and their relationship to disease pathologies. And for those of us that are on Twitter, Cara is also doing a lot of public engagement. So it's really great to have you today. And the stage is yours.
Thanks, Mahmoud, and thank you for everyone who's joining us. There's already been some really great talks, and I'm excited for the rest of the session. So today, I'll be talking to you about neurofibrillary tau inclusions, and as Mahmoud said, I'm always happy to answer questions either by email or on Twitter. So you can see my email details and handle there. So I'd like to start by saying that this work is only possible because of a really great team that have supported me through my career to date, and of course our wonderful funding sources. And most of the data that you see today has been generated because of a Bright Focus Foundation Fellowship and a Race Against Dementia Fellowship, as well as wonderful people that have worked across these projects. So I understand that this is a neurodegeneration meeting, and most people will know a little bit about Alzheimer's disease and other tauopathies, but I will give you an introduction here. So tauopathies are neurodegenerative diseases leading to massive neuronal loss progressively over time. Tauopathies are so called because you find inclusions of tau, normally comprised of misfolded or phosphorylated tau protein, in cells of the brain. So not just neurons, but also other cell types depending on the type of tauopathy, such as Alzheimer's disease, frontotemporal dementia, progressive supranuclear palsy, as well as other diseases affecting millions of people worldwide. So there are a large spectrum of symptoms, including memory problems, communication problems, other cognitive issues and motor problems. And one of the main things is that the accumulation of tau has been linked to neuronal loss and cognitive decline in Alzheimer's disease. There are currently no cures for these fatal diseases. And similarly, there's been limited understanding of how the buildup of these tau inclusions and what species of tau actually link to cell dysfunction and death. And here you see some thioflavin S staining of neurofibrillary tangles in an Alzheimer's disease brain. So back in the 90s, several mutations started to be identified in the gene microtubule associated protein tau, which produces the tau protein. And on the basis of this, these mutations have been used to develop models. So transgenic mice lines expressing some of these mutations are widely used in the field, as well as cell-based assays, and more recently, iPSCs derived from some of these lines. So the start of this work was with Kevin Strang in Ben Gierson's lab at the University of Florida, and he used a HEK293T cell-based seeding assay and screened a lot of those MAPT mutations to see whether they could seed. But the main finding was that only a couple of them actually robustly seeded. But what he also found was that P301L in combination with S320F was able to self-aggregate, and this is in the space of 24-48 hours. You get massive amounts of aggregation without the need for seeding. So a lot of my experience from my PhD was the preparation of organotypic brain slice cultures from transgenic mice, but then I moved to Todd's lab to learn more about AAVs, using those with in vivo models, and similarly employing those in the organotypic brain slice cultures. So there's several advantages of AAVs. We have now worked out various different capsid and promoter combinations where we can target all the major central nervous system cell types. In a slice culture model, they express rapidly, so 4 to 7 days, you start seeing massive expression, and this is sustained. So out 12 months in slice cultures, we can still see what we expressed. Co-transduction is possible, so you can study the interaction of different genes that you want to overexpress. And similarly to kind of transfection regimes, you can do easy parallel studies of several mutations of your favorite gene, or screen several things at the same time, which is typically not possible with transgenic mice, where you'd have to have lots of different colonies, and that can get expensive. So organotypic brain slice cultures, they have several 3Rs benefits. So from a postnatal day 8 mouse brain, you get 12 to 36 experimental variables. They retain the cytoarchitecture and the central nervous system cell types. So in these cultures, we have microglia, we have astrocytes, we have oligodendrocytes and neurons. So we can start looking at the relationship between these cells as we're modeling disease. And they remain functional for a long time in culture and survive a long time. And particularly for the data that you'll see today, you can easily manipulate the environment. So for imaging, or the addition of AAVs or drugs, you can easily do that in these models. And if you want to read more, this is just a review that I published last year, which hopefully gives you some of the advantages. But as always, I think as we're studying neurodegeneration, we need to be using multiple models to really get at what is going on in disease. But these provide a nice platform bridging cell studies to in vivo or human studies. So back in 2019, we worked on the basis that the fact that P301L in combination with S320F aggregated rapidly in the HEK cell assays with Kevin Strang and Ben Gierson. So we developed AAV constructs expressing the P301L S320F tau and transduced organotypic brain slice cultures and did a thorough characterization of these models. So at this point, I think the main things to highlight that when we overexpress this P301L S320F tau, we get rapid progressive development of tau inclusions. So 7 to 14 days, we're already seeing an abundance of tau inclusions in these slice cultures. And importantly, tau inclusions exist for a long time in these slice cultures. So you can see here, out at 90 days, these inclusions still exist. So there's a large window to study them. And similarly to some lines of evidence where these inclusions can be in the human brain for 15 or more years, they are long existing structures, and we can model that in this system. So moving on from this, this is all unpublished data that I'll show you now. We've been using tau dendra, which is, dendra is a photo-switchable fluorescent protein. So it starts off as green, and then you use blue light photo conversion, and you can then track any old protein in red and newly synthesized protein in green in these optical pulse labeling experiments, which are really easy because the slice cultures are really accessible for imaging. So we believe these have a really good utility for understanding tau clearance and production. We hope that they will start to allow us to begin to understand a bit more about immunotherapies or small molecule compounds and their effects on tau, as well as determining other gene effects on tau dynamics, which is a part of my Race Against Dementia Fellowship, as well as being able to long-term track these individual cells with tau inclusions. So the next part of my talk will just be about the formation of tau inclusions, because obviously hand in hand with microscopy, these cultures are easy to watch and we can start trying to see the formation of tau inclusions. However, we wanted to control this. So in the field, the use of P301L tau with the addition of seeds to stimulate the formulation of aggregates is a way of controlling exactly when the aggregate starts to form, compared to our previous studies where we get this rapid formation. So you can see here when we characterize that the addition of tau fibrils in the space of 10 days, we do get this shift to a lot of phosphorylated and insoluble tau accumulating. And again, when we do the thiazine red quantification, most cells that have been seeded bear aggregates by 10 days. So we have a window there of 10 days where we know that cells don't have aggregates, then they do have aggregates in the period of the addition of the fibrils. So here we add the fibrils, we photoconvert immediately after the addition of fibrils and track them. And then in the next part, we photoconvert again, we can image their stability and clearance. So here, this is just the P301L tau dendra where we didn't add the seeds, and you can see that there's tau, and then the tau declines with a half-life of around 3 days. Then we photoconvert again, and you see the same again. Here, now we've added the fibrils at this point, and you can see that some of this already photoconverted tau starts to form into aggregates in the cells in these brain slice cultures. So we have started to image the formation of the inclusion here. And then when we repeat photoconvert, you can see that this is a stable inclusion that is formed. Now, This again shows that seeded tau inclusions are dynamic structures with a constant but slow turnover. I'm not showing you that data, but essentially the half-life is around 7 days. And just because of time, I can't show you that, but it seems to show a similar story, that whether your aggregate and inclusion forms intrinsically or with the addition of extrinsic seeds, they still are dynamic structures with a constant but slow turnover. And then lastly, here we used the MAP2 promoter, and we were able to just more slowly track these individual cells with the self-aggregating tau mutations. And you can see within 24 hours, you have this somatic accumulation of tau, yet this exists for a long time after. And then we have another population of cells where the aggregate tends to form over a few days. But essentially, this work has been done before in vivo models with 2-photon imaging, but this is just an easy platform to start getting at what causes the formation of tau inclusions. So today I'll just summarize and hope I've shown you that there are several advantages of using AAVs and brain slices to study tau, but of course these are complementary to all the different methods out there that we can use, and just start to reveal some of the story. Using Dendra2, we are performing these optical pulse labeling experiments, which enables us to understand the dynamics of tau protein and track individual cells with and without aggregates over time. We can easily visualize the formation of tau inclusions, recapitulating previous in vivo data, where similarly in vivo in mice, they form in a 24 to 48 hour period when you are watching them using 2-photon imaging. And we also show that tau inclusions are dynamic structures with a constant but slow turnover. And this is important because maybe understanding these processes which mediate this turnover may lead, or even accelerate this turnover, may lead to novel therapeutic interventions. So just a couple of plugs at the end. The CTRND has been an amazing place to be a postdoctoral fellow, and there are always postdoc and PhD student opportunities available. So I'd suggest to check them out if you are looking for your next position, as well as the Race Against Dementia Fellowship. This is £500,000 over 5 years if you're an early career researcher, so within 3 years of finishing your PhD. And applications are open now, and I think they're recruiting 3 new fellows. So it'd be important to start thinking about a fellowship if you are at that stage of your career. And I did thank everybody at the start, but I'll always thank them again. And I'd be happy to take any questions in this discussion session or by email or Twitter. So thank you.
Thank you very much, Cara, for a wonderful talk. Really interesting stuff. Can I kind of remind all attendees that they should send their questions so that it can be discussed at the end of the presentations. So now we'll be going to the last ECR talk of the day, which is going to be given by Dr. Oliver Bracko. Before I introduce him, can you share your screen while I do that? So Oliver will be talking to us about, okay, let me just go back there. Okay. So Oliver will be talking to us about inhibition of peripheral VEGF signaling. Inhibition of peripheral VEGF signaling rapidly reduces leukocyte obstruction in brain capillaries and improves cortical blood flow in an Alzheimer's disease mouse model. And Oliver was trained as pastry chef before studying at the University of Tübingen in Germany. He pursued a master's at the Salk Institute in Rusty Gage Lab in California in the United States, with a fellowship from the DAT, which is a fellowship from the German government. He followed that with a PhD in neuroscience at the ETH Zurich in the lab of Sebastian Jesberger on the molecular signature of adult neuronal stem cells. He then joined the lab of Susanne Wegner at the University Hospital Zurich, Switzerland, as a postdoc, where he worked on the cause of post-stroke cognitive impairment. Oliver later secured a postdoc fellowship from the German Research Foundation in Chris Schaffer's lab, working on the role of brain blood flow reduction in Alzheimer's disease. He's a recipient of the Postdoc Achievement Award for Excellence in Teaching and Mentoring from the Cornell University. Oliver, you are highly welcome, and we all look forward to your exciting talk.
Thank you, Mahmoud, for the nice introduction, and thank you for giving me the opportunity to present our data here today, which is under the umbrella of our efforts to understand the role of brain blood flow reductions in Alzheimer's disease. And in particular, I'm going to talk about a story that has been developing over the last 2-3 years, where we're trying to understand the mechanism of this phenomenon, and we're focusing here on the role of VEGF signaling to brain blood flow reductions in mouse models of Alzheimer's disease. First, I want to thank Chris Schaffer and Nozomi Shimura for mentoring me over the last couple of years, and then of course all the people who are involved in this or previous studies, but in particular the student is Muhammad Ali, who developed this story, and he came up with the idea at the beginning. So I really have to thank him. He was my first undergraduate working for me through the last 4 years, and of course our collaborators and also our funding agencies. I don't need to talk about the importance of Alzheimer's research because we heard that from the other speakers already. So I really want to focus on our topic. We were interested why there's baseline blood flow reduction seen in patients and also in mouse models of Alzheimer's disease. Now, it is known since a long time that there is reduced blood flow, and you can see that in the study here from Aslani et al. From 2008, where they looked at arterial spin labeling MRI to measure blood perfusion, so to measure blood flow, whereas light colors are high perfusion, dark colors are low perfusion. And you can see that in the control patients, you can see throughout these cortical structures, there's high perfusion, meaning a good or healthy blood flow. But if you look in Alzheimer patients, you see there are much more dark regions. They speak for reduced blood flow. You can also see that in this difference map. So in humans, it can be about 10 to 30% overall. And this is also reflected in mouse models of Alzheimer's disease. And of course, we were at the beginning just interested, what is the role or what is the contribution of this blood flow reduction to the overall disease progression? So we are an engineering lab, so we are building multiphoton microscopes. So we're using 2-photon microscopy for this study today, where we have chronic cranial windows in mice. So we replace the skull with a glass window to get optical access to image those mice in vivo. So usually we do that 3 weeks before we image the mice, just to get rid of this topical inflammation. And on the right here, you can see a typical picture that we get from such an imaging session, where we label the vasculature with a dye is called a Texas red bound to a dextran, in that case in gray. So it labels the blood plasma, but it doesn't label the blood cells. And in red, you can see, this is an APP-PS1 mice, you can see the plaques labeled with methoxy. So this mouse is a little bit tilted. You can see the large vessels, the arterioles, and the capillaries. So we were looking for changes in the veins and the arteries where this blood flow deficit may occur, but we didn't see differences. So we really looked into the smallest vessels, the capillaries, and this is where we found the phenomenon that we called stalled capillaries. And you can see that here in these pictures. So this is the capillaries, and you have to keep in mind that the capillary diameter is basically the same as the cell diameter. So you see one cell flowing by in such a capillary, and you can see that here. So the plasma is labeled in red, and the black spots are the blood cells. So they're not labeled, they appear black, but what you can nicely see with time, you see the black spots moving along this capillary, indicating blood flow. However, what we identified that a certain percentage of those capillaries are stalled, and you can see that here, that means with time they are not flowing. And we quantified that first in wild-type mice. So this is a fraction of capillaries with stalled blood flow. So it's around 0.4%. But if you look in APP-PS1 mouse and also in other mouse models, we see it's around 1.8% of all capillaries are stalled at a given time point. So we call it stalled because it's only transient. It's usually from a few seconds to a few minutes, and then the vessel is flowing again. So we published this work last year. I just want to add 2 more slides to show you what are those black spots, so which blood cells are responsible for most of the stores. And we did this in vivo labeling strategy, where you can see here we label leukocytes with rhodamine and DNA host, which is a DNA marker and a mitochondrial marker. And then you can see the platelets, which only have the rhodamine labeling, and the red blood cells are black. So when we quantify that, we could show that around 60% of all stores are formed by leukocytes and around 30% are formed by red blood cells. Now, of course, we were interested which cell type within the class of leukocyte is responsible for this. So we did another in vivo labeling strategy and just started with the most abundant leukocyte, which are the neutrophils. So we stained it with Ly6G. This is a mouse-specific antibody against neutrophils. We stained it, and we could show that around 90% of these capillary stores are formed by neutrophils. Now, what we also saw at the same time then when we injected Ly6G, we also saw a rapid reduction in the number of capillary stores. So within 10 minutes after injection, all the capillary stores were gone. And of course, this was really exciting for us because this allows us to do now manipulation and also do further studies. So I just want to show you one example where we measure the blood flow after such a treatment. This we did in collaboration with Liebach Park from Weill Cornell Medical College. And this is again arteriospin labeling MRI, where the light colors are higher perfusion and the dark blue colors are low perfusion. So if you look at the wild type mice, you can see there's cortical perfusion also after treatment with the antibody. If you look at the Alzheimer mice, you see there's low cortical perfusion. And also if you use an ISO control antibody, which means it's a non-targeting antibody, you can see there's low perfusion, but if you inject it with the Ly6G mice, you see recovery of blood flow in those mice within about 2 hours. So we quantified that here on the right. You can see the overall blood flow in this Alzheimer mice, and you can see here on the right, and when we inject Ly6G, roughly 2/3 of this blood flow can get restored. To summarize this previously study is if we have stored capillaries, as you can see it here, mostly formed by leukocytes, but not only, you can see there's stalled blood flow, and the stalled blood flow contribute to blood flow reductions. And if we inject Ly6G antibody, we see improved blood flow. I haven't shown you, we see improved cognition, and we see a reduction in amyloid beta burden. And we don't see that if we use a control antibody. And now the question was, of course, what is the mechanism leading to this phenomenon? And we were thinking about, of course, there's a couple of theories we are following up. One of them, is there change in deformability or in stiffness of those leukocytes? Why is they having more trouble to squeeze through the vessels? Or is there increased vascular inflammation, which also lead to increased sticking of the cells? Or what I want to show you today is the role of the blood-brain barrier. So is the blood-brain barrier more leaky, and this attracts the neutrophils and causes those capillary stores? Just a few words about VEGF. It's broadly impacted in Alzheimer's disease, and it can act via many signaling pathways. But overall, it gets produced in almost all cells. It gets secreted into the blood vessels, but also into the lymphatics and also into the lymphatics. But what was really exciting for us, there has been a study from Patrick Greason here from Craig Brown's lab in 2018, where they inhibit VEGF2 receptor, and then they saw improved clearance of microsphere-induced cortical capillaries. And of course, that was a good hint for us that maybe in Alzheimer, it could be similar. And when you inject VEGF directly into the brain of a mouse, you can improve short-term memory, and you can also reduce the amyloid beta burden. So just once again, so this project has been developed also with Muhammad Ali. He has been an undergrad student for me, but now is at med school in Mount Sinai. So just one more word about VEGF and how it's contributed to blood flow reduction. So you have all these cardiovascular risk factors and also other factors that contribute to brain blood flow reduction. You have then hypoxia, and that leads to increased VEGF. However, in Alzheimer's, it's known there's this angiogenic phenotype where you have increased VEGF, but you don't have the signaling anymore. And now we thought maybe this somehow contributes to our capillary stalling phenomenon. So first, We looked again whether we can repeat our phenotype, and indeed this is the case. So these are stacks. So these are 2 photon imaging stacks from wild type mice and from Alzheimer mice, whereas the stalled capillaries are in yellow. And when we quantify that, as you have seen it before, so in Alzheimer mice, we really have an increased number of these capillary stalls. And also the cell types is similar. So we see most of them are leukocytes, and the second largest fraction are red blood cells. Now the study, we had 4 groups. We have an Alzheimer group, we have a wild type group, with and without anti-VEGF antibody. And the way we performed the experiment, we did the craniotomies, we waited for 2 weeks, then we started to inject anti-VEGF antibody into the mice, imaged after 1 week, continued the treatment, imaged after 2 weeks, and then we sacrificed the tissue. Of course, the first thing we were looking at is there a change in the number of capillary stalls? And indeed, that is the case. You can see that on that graph on the y-axis, you have the fraction of capillary stalls, and you can see in Alzheimer mice that only got a saline injection after 1 week or after 2 weeks, so there's increased levels of capillary stalls. But if you inhibit VEGF with an antibody, you see a reduction in the number of capillary stalls, basically to the wild type levels. And of course, next we also measured the blood flow, this time with another technique, which is a line scan, where you use the 2-photon microscope to image individual vessels, the speed. And this is a quantification here on the right. So it's the vessel speed on the y-axis, and you have the saline injected APP mice. So the Alzheimer mice, you can see there's reduced blood flow, but if you inject anti-VEGF antibody, you see a restoration of this blood flow. So there's improved blood flow, and that remains elevated until 2 weeks. If you look at the vessel diameter, you can see there's no change in vessel diameter, indicating us that vasoconstriction is probably not involved in this phenomenon, but that needs to be determined, because there's been a recent study from David Edwell's lab, where he has shown that pericytes constrict as capillaries and probably also contribute to blood flow reductions. We have to see how that may merge with our story. What is the mechanism? So VEGF has a broad spectrum of downstream signaling. So we were screening the literature, and we were trying to identify a pathway that leads us to the blood-brain barrier. And we were looking into this eNOS occludin pathway. The main reason is because it's really acting fast, at least in cell culture. And first, what you can see here is an ELISA measuring the VEGF concentration from brain lysates. And what you can see when we inhibit VEGF, you really see a reduction in the levels of VEGF in those Alzheimer mice and also in the wild-type mice. So next, we tested then the eNOS level. So that's the endothelial form of eNOS, which leads to NO signaling, but it also has a direct effect on the blood-brain barrier. So this eNOS is also reduced in those lysates. So we really have a reduction of eNOS after treatment of the mice. And then we stain for blood-brain barrier proteins to see whether we see changes in permeability. And you can see that here. So occludin is a tight junction protein. It's known to be directly regulated by VEGF. You can see that in green, and in red you have the vessel marker GLUT1. It's glucose transporter 1. So what you can see here is basically an overlap between those two stainings. So in the wild-type mice, we have a high expression of occludin, basically at all levels, indicating an intact blood-brain barrier. If we look at the Alzheimer mice, you can see there's hardly any occludin, indicating there's a leaky blood-brain barrier. So if we inject those mice with anti-VEGF for 2 weeks, you can see here on the right that we restore this occludin levels, indicating that we are closing the blood-brain barrier. We quantified that here on the right. So we have APP mice. At baseline, you see there's not much occludin signaling, but if you inhibit the anti-VEGF, if you inhibit VEGF, you see a restoration of this protein in the cortex and also in the hippocampus. Now, we were really interested whether is there a direct change of occludin levels at individual capillaries that are stalled or flowing. So for that, we took on the challenge to really look into our in vivo 2-photon data. And as you can see it here, and try to identify the same positions in the histology sections. And for some capillaries, we were able to do that. You can see that here. So this is, of course, the same position. And we have here stalled capillary, and the other ones have been flowing. When we quantified that, we could show that at the ones that are stalled, we have reduced occludin levels, and the ones which are flowing, we have more occludin. So that's really for us the first hint that maybe something is special about the capillaries that are stalled at a given time. To finish it, we also looked into Evans blue. So Evans blue is a dye that diffuses into the tissue, into the parenchyma, and it can be used as a measure of blood-brain barrier leakage. So what we did, we injected mice with an anti-VEGF antibody and Evans blue, and 1 hour later we killed the mice and looked for leakage. And what you can nicely see here, this is cortical absorbance, and what you can see in a saline injected mice, you have high parenchyma Evans blue signaling, but if you have an anti-VEGF treatment, you see it reduced basically to the right wild type levels, indicating us that the blood-brain barrier is really closing. And the same you can see in the hippocampus, but it's not significant. It's just a high variance in the sample here. To summarize this, what I've shown you is that we in APPPS1 mice, we have these stalled capillaries, mostly formed by neutrophils, which block individual capillaries and contribute to the overall blood flow reduction. We have increased levels of VEGF-A, which has a downstream signaling to the blood-brain barrier, which is leaky in Alzheimer mice. Now, when we look into the anti-VEGF-A treated mice, we can see that we sequester away the VEGF, we reduce the number of capillary stalls, we restore blood flow, and we close the blood-brain barrier, indicating us that there's a direct link of those capillary stalls and VEGF signaling. Of course, there are many open questions we need to follow up. So which is the receptor that really mediates the signaling? What is attracting those neutrophils to the leaky blood-brain barrier? It could be many possibilities. It could be, of course, surface receptors. It could be the amyloid beta itself, which get also leaked into the blood at sites of blood-brain barrier breakdown. Thank you for your attention.
Fantastic. Thank you very much, Oliver. It's such a great talk. I really have to thank Rebecca and ProteinTech for organizing this meeting. It has been so great. I mean, all the different talks. I think this is something that should be organized almost every year. I think we've got about 30 minutes for Q&A, so which is quite good because all the speakers would have the opportunity to respond to all the questions that you've sent. Without wasting time, and since we started with Henriette, I guess it's only fair to start the Q&A with her. So we've got a question from Kartik, and she said, what is the earliest time point in culture when you start noticing mitochondrial and Golgi defects in patient-derived neurons?
So we have, I mean, most of these analyses have been done after 7 weeks of maturation, but we are currently looking into 5 weeks old neurons, where we also see, especially the Golgi fragmentation already at this time point. Of course, in terms of synapses, we are only able to see it after 7 weeks because of the maturity level of these neurons. But with the other organelle pathology, Golgi fragmentation seems to be the first pathology that we are able to observe, which is also fitting with our hypothesis that this might be the first step of this, and then further leading to the processing issues of other proteins.
Okay, that's quite interesting. I'll come back to that point that you raised, but I guess let's move on to Siobhan to hear this question first. So there's this question from Tara to you, Siobhan. She said, does the high neurodegeneration potential of zebrafish cause any confounds in your stroke studies?
This is an excellent question, Tara, and something that I deliberately left out of our talk. So zebrafish do regenerate, and the recovery from the intracerebral hemorrhage in our model is very quick as well. We don't keep our model past 5 days, but we have noticed that the hematoma clears by day 4, and the resolution of that brain cell death is clearing again by day 5. So we don't think it is a confounder. I think it's actually quite exciting that these zebrafish do fully regenerate from a brain hemorrhage in larval stages, and they go on to become viable adults that we use for breeding all the time. So we're also interested in looking in that further down the line and see if there's some of those regenerative pathways could be activated in aged people in order to recover from a hemorrhage better and save those cells that can be used for pathways.
That's quite interesting. Thank you. Cara, we've got this question which I had in mind as well, but it's good that someone asked it. So it said, do you know what regulates the tau turnover? Is it enzymatic? Does tau localize to lysosomes, autophagosomes? What actually does regulate the turnover?
So I think this is the one that will be the next few years' work. There's been evidence essentially for tau degradation by autophagy, tau degradation by the UPS, tau clearance by astrocytes, tau clearance by microglia, and I think any of those are possible. Whether one is more dominant than the others, I don't think it's clear, but I think any of those are definitely possible and quite easy to study in this system as well as other systems. So I think that's what we need to start getting at, because definitely after that, if you know stimulating autophagy is the way to go, that's probably a therapeutic route that would want to be considered.
Yeah, absolutely, I agree. So hopefully with more money, you'll be able to address that in the coming years. All right, so let's move on to Oliver. So what I'm trying to do is try to distribute the questions across all our different speakers, but of course you can continue to send in your questions and we'll try to address them. So Oliver, there's this question that said, would there be disadvantages to lyse 6G antibody to other parts of the body? So would it have any problem to other parts of the body?
Yeah, it also has a problem overall. So Ly6G antibody, and I left this published part out in the sense of, so within 1 day you deplete your whole neutrophils. So finally Ly6G antibody leads to depletion of all neutrophils, and of course that compromises the whole immune system, so it has an effect on the whole body. The next problem is that this Ly6G is also a mouse-specific protein. It doesn't really exist in a known homologue well in human patients. So this is a little bit tricky, but it's just the best, purest marker for neutrophils in mice. So that is why we have used it. And of course, we were also surprised seeing this phenotype. But we know that we have two mechanisms in play. We have an early phenotype where we see that somehow the adhesion goes away because no depletion occurs within the first few hours, where we see this rapid reduction in the number of capillary stalls. But later on, the blood flow remains reduced, and it's correlated to this number of stalls. But if you deplete the neutrophils, it just remains always up, the blood flow. So there are two effects in play, and I think it's quite complicated, and we haven't deeply studied late effects of depletion of those neutrophils. But there are some other studies from another lab, from Gabriele Constantine in Verona. She has used also anti-neutrophil antibodies, and has shown even on the long-term effect that this leads to, she didn't measure the blood flow, but it leads to improved cognition that remains quite long while she's treating those mice. So I think it has a long-lasting effect, but it depletes finally the neutrophils. So of course, that is what we don't want in a patient.
Yeah, absolutely. And I was wondering, although it might sound naive, but did you look in to see if you have an improvement in some of the pathologies that you see in that mice model when you administer these antibodies?
Yeah, I left. So actually not, yeah. So yes, we did look at it, and there are some changes. So but it was less than we thought it would happen. So we treated this mice and for a long time with this antibody, and we definitely see a reduction in amyloid beta. And we did that in histology, we did ELISAs, but we only saw a reduction in the monomeric species, the A40. And then I was also excited about it because I know that the A40 is more associated with the vasculature. And, but then I tried to really look into it. I tried to see whether CEA plaques are more affected with the treatment, but I just didn't get the result. So there was just no difference. So all we had was this reduction in this A40 species and everything else remains the same. But we still saw the increase in blood flow and we saw that these mice have improvements in spatial memory. And I mean, those mice have an overexpression of amyloid beta, so there must be a connection. I just haven't seen it yet.
Absolutely, absolutely. Let me come back to you, Cara. So there's a question from Sarah. She said, have you tried to check if you get the same findings if you made slices from P301L pups instead of AAV transfection?
I've not tried that, but I think it's likely that you would probably see similar. I think it's that P301L seeds well, and whether that's through a transgenic mouse or through AAV. I mean, we prefer AAV because you can somewhat control the level of expression. So if we're doing different mutations in parallel, they're expressed at a similar level, whereas transgenic lines, you never really know how much fold over you're gonna get. And I do think I've seen possibly at a couple of conferences, people have used P301L tau transgenics and seeded them, and they've got seeding. So you could do it that way if you were interested in studying seeding that way.
I see, absolutely, absolutely. To our attendees, you can still send questions. We've got quite a good time to answer them, hopefully. Coming back to Siobhan, there are about 3 questions from Marietta, but I'll probably just ask one and then I'll get to ask the other one later on in the second round. So the question basically from her is that I'm wondering, in terms of translation of the drugs, whether you consider that targeting neuroprotection by itself may not be sufficient for successful therapeutic intervention. Have you, can you tell us a little bit about that?
Yeah, so hi Henriette, and it's nice to hear from you. So we aren't exclusively looking at neuronal population in this annexin 5 region of dying cells. For morphological analysis, we can see that there are glial cells in there and sometimes whole areas of endothelial cells making up vessels as well. So if we can protect that region of brain, then we've shown that that results in a functional beneficial output, and that's what we want for our patients. Additionally to that, we know that preserving neuronal function has a downstream effect on preserving glial function as well. So if that's the strategy that we can work out the mechanism for ACE inhibitors, then it's good enough for us. Alternatively, this is really new, so we've only just identified these ACE inhibitors from our drug screen, and we haven't had any further investigation. This is preliminary data that I've been showing you today. So in order to identify what exact mechanisms ACE inhibitors are exhibiting neuroprotection, we haven't identified yet. So it might be a cell type wide mechanism. There's quite a few papers to show that ACE inhibitor neuroprotection comes through protection of mitochondrial function, which would also have a downstream effect on glial cells.
I see. But let me follow that up with the same question that she asked. She said, have you identified drugs that you, did you move any of your identified drugs to mouse or large animal models of brain hemorrhage?
No. So if anyone knows me, I like fish. So we only model in fish in our lab, and we haven't had a chance yet to look at any other models. The idea is that we can use this for translational research, and if we get enough evidence of repurposability in the clinic, then we can avoid that entirely. So.
Absolutely. All right. Let me come back to you, Henriette. There is this question about the role of microglia in affecting the process of disease in AD. So I guess the question is probably, if I understood it correctly, whether you've considered the role of microglia, but I guess your culture model is just the iPSC, you are not co-culturing But anyway, can you tell us a little bit more about that?
Yeah, this is something we are working on at the moment. So right now we have, from these cell lines, we have the monolayer of the neurons only in culture, but we are currently working on differentiating microglia. So then as a starting point, we will use the supernatant from these microglia to then add to the neurons to see the effect of microglia on these neuronal phenotypes. And then the next step would be to then establish a co-culture. So then we can see if, for instance, the microglia in the deceased cases would then be more detrimental to the neurons compared to the control cases. And this, we're also doing this with sporadic AD at the moment to get a full neuroinflammatory panel of AD.
Okay. Well, I guess there's this question which is supposed to be the fourth one, but it fits in quite well with what you just mentioned, sporadic AD. So the questionnaire asks, what are the challenges in using iTesting for sporadic Alzheimer's disease modeling?
Yeah, so that is, as I mentioned also in my talk, the genetic profile, because FID is very easy to then, if you use CRISPR-Cas9 to generate matching controls, you cannot do this with the sporadic AD cases, most likely, because you don't necessarily know the exact, you don't have one disease-causing mutation that you can go in and correct in most cases. So that makes it challenging to then provide a healthy control, because then the control you would have a different genetic background. So in this way, it's more challenging with the sporadic AD modeling.
Yeah, I see. Yeah, let me just jump back to Oliver now. So there's a question that said, I'm wondering, are there more leukocytes or neutrophils in capillaries of transgenic mice?
So no, so in mice, we don't see an increased number of leukocytes overall, but in patients, and in a good fraction of patients, you see increased levels of leukocytes/neutrophils in patients with Alzheimer's disease. So this mouse model doesn't really reflect that as well. So it's not really the numbers. We've done flow cytometry and had a look whether is the starting number really different compared to wild-type mice, but this just wasn't the case. That does still mean it could well be that there's also some indications that, of course, not all the neutrophils or all the neutrophils are the same. There might be also subpopulations of them existing that may explain such a phenotype.
I was wondering, but what do you think about the next step of your work? I mean, you are now using this mice model. Are you thinking about trying these antibodies on different models, or what are you thinking about?
I mean, yes. So by now we know that those capillary stores exist under several conditions. For example, if you do a stroke, you see a massive increase. If you do a TBI, you see it, and you also see it in other neurodegenerative diseases. But that doesn't really answer your question. So I would say, so of course, the most important thing we would like to know is, does those capillary stalling also exist in humans? That is something we don't know. And then the reason is we just don't get the imaging resolution to really look at those capillaries. But as technology is catching up, there probably will be soon some ways to do that. And there's some effort from other labs trying to use a multiphoto microscopy in patients to image the retina of the eye and look at the capillary bed there. And that might be a window for us as well to see whether those are really existing in patients. And of course, that would be crucial for us to kind of more justify all the work. But at the current stage, we cannot really do it. You could imagine doing an indirect study where you would have a drug that interferes with leukocytes, and there are some drugs which are FDA approved, and you would test whether you see an increase in blood flow if you use them, and that would be an indirect measurement. But this is really tough to justify just to get the approval for doing something like it if we don't really have the data. And the problem is Ly6G just doesn't exist in humans.
Yeah. Just remind the attendees that our plenary talk of the day would be given by Professor Cara Spires-Jones, which is probably going to start in the next 10, 15 minutes. So you should definitely get your coffee or tea and come back before then. Yeah, so moving on, let's come back to Cara. So there's a question from Sarah. It said, can you explain more what you mean by turnover? Is it death of hosting neurons or microglia?
No. So these are living cells, and that's what's been good about using this method to study it, that we know that the cell at the end is producing new tau and it's a living cell. So it's the tau is being cleared by some sort of mechanism that we don't know yet. I answered that question earlier, that it could be autophagy, it could be UPS degradation, but something is going on with the cell which allows these aggregates to get cleared, and whether speeding that process up is the way to go, we don't know.
Okay, cool. But there's this question from Tara. She said, what do you think about the relationship between seeding inclusions and spreading pathology between cells? Do you think it's always aggregate-prone versions of tau that also spread through the brain?
I think there's evidence for pretty much every single tau species spreading through the brain. And I think one of the issues in the tau field is that everyone has their favorite flavor of tau, where they try and say it's this. And I think probably they all contribute in some way. But one thing I would say is that obviously in this model, we can see, I have just a small area of the section where we have photo-converted tau. I don't necessarily see that tau spread through the culture to other areas. So in this model, it's just aggregation within those cells that I'm seeing. No spread.
I see. So perhaps if it was a different mutation, or do you think that you could see, do you think this is specific to this particular mutation that you're looking at?
Possibly, yes. I think a lot of the seeding studies have been done only in P301L mice, and there's a little bit of evidence that this can happen in non-transgenic mice if the conditions are right. But I think a lot of the seeding studies are always dependent on certain conditions.
Okay, cool. Okay, let me move on now to Siobhan. So there's a question from Marietta again. She said, we start to understand the mechanisms by which other cells of the brain die in response to brain hemorrhage, and that this may be different and hence will require a multimodal therapeutic strategy targeting different cells. Can you somehow model this in your zebrafish model and maybe also screen the positive hits for their function in other cell types or test combinatorial approaches?
Yeah, sorry, Mahmoud, I think I probably answered this in the first response as well. So where we see a neuroprotective response, we're not protective response, we're not entirely limiting to neuronal cells. But Marietta raises a really good point. We could be culturing cells in different cultures of cells and test the ACE inhibitors in those particular cultures to look at protection and durability. There are all sorts of ways to screen for this. From our point of view, we're looking at more of a functional, overall kind of holistic approach. And if we can translate those drugs, it's even better. So that's why we've limited it to ACE inhibitors, not necessarily because it's neuroprotective.
Yeah, okay. And James was wondering, he said there has been some discussions regarding COVID-19 and ACE inhibitors. Is this something you have looked into, and does this have any relevance towards stroke?
Yeah, so hi, JQ, it's nice to hear from you too. Obviously, ACE inhibitors have been shown to be beneficial in COVID, because if you stop taking them, then you're going to have a detriment. ACE2 is the protein that the COVID-19 needs to enter lung tissue. And from a ACE inhibitor mechanistic point of view, it's probably due to inhibiting the expression and the function of that enzyme on the cell surface, rather than from an antihypertensive approach or from a neuroprotective approach. You're right, COVID-19 has been associated with stroke as well, and I think it's fair to say that we don't really understand much about the pathology in COVID-19.
Yeah, absolutely. All right, let me now move on to Henriette, and I think these couple of questions are all regarding the synapse and some of the things that you've mentioned. So there's a question from Claire. He said, I was wondering if you have done any analysis of the co-localization of synaptic markers like PSD95 and synaptophysin.
Yeah, so this is something we have done also some experiments with the antibody for PSD95. And I have left this out in this presentation because we've had some issues with this antibody in terms of it being unspecific. So that's why I can't draw any conclusive or can't draw any conclusions based on these stainings. That's why we're currently also doing this in the lab. So for now, we only have the synaptophysin data validated.
Okay, so I guess this is something that you, because there's this question from Soraya who was saying she's trying to understand whether you are seeing less synapses or less vesicles at the synapses. And I also wanted to actually ask about that, because you might probably be having less synapses, but does it really mean the functional level of this of those synapses that are remaining is distorted, since we've got the recycling processes happening, etc. So I was wondering whether you are thinking about looking at, or whether you've looked at that.
Not in terms of the functionality, no. So basically, because we can see both in the TEM, we see both a reduction in the amount of synapses, but also in the synaptic vesicles. And then the use of synaptophysin, that's also a marker of the presynaptic vesicles. So we can for sure say that we have a reduction of these vesicles. But in terms of the function, we don't have functional analysis of this at the moment.
Okay, cool. All right, I think now I'll probably got two questions for, one question for Cara and Siobhan before we probably end this session. So I was wondering, Siobhan, if you can, because I'm interested in any model that would simplify research in Africa that I advocate for. I was wondering if you can briefly tell us, how expensive is it to set up like zebrafish model system like you do in your research?
Yeah, so I only work with the larvae. So we have breeding tanks of many transgenic lines and genetic mutant lines, which aren't always necessary. For this one particular model, bubblehead can be bred from heterozygous parents and have a homozygous offspring, which will hemorrhage, and the heterozygous and wild type offspring are negative for hemorrhage. So you can get your control population from the same clutch of animals, which means you don't have to breed one genotype of tank. The experimentation is all very quick. As I said, only takes 5 days between fertilization and when we terminate our experiments. So I think it is probably quite accessible. We do collaborate with the University of Cape Town and to help them with some of their cardiovascular drug screening and assays that they're doing, that they can do the equivalent in a zebrafish much quicker than they could do in a mouse model. And this is something we're very enthusiastic about. I think that there's a lot of potential that is untapped in a zebrafish model, and always looking to collaborate with other labs.
Fantastic. Well, I follow you on Twitter, so hopefully you'll be tweeting about new developments so that we can all know what's happening. Okay, I think this would probably be the last question of this session, and it goes to Cara. It's from James Quinn. Hey, James. James said, the half-life of tau in the human brain has been recently published. How does the half-life tau turnover compare between the mouse cultures and human brain?
So I think he's referring to the WashU study in 2018, where they looked at iPS cells as well as in human CSF. And the way they were doing it, they were looking at different phosphoepitopes. So they showed that different regions of tau and different phosphoepitopes have different levels of turnover. But what we're looking at is the aggregates. So I think no one's really shown the half-life of an aggregate before. What we found is that at these early stages, once the aggregate has formed, that the half-life is around 7 days. But once these have aged for longer times in culture, that half-life is getting longer. Back to that paper, they show in iPSCs, I think the half-life of most of the species was around a week, whereas in the CSF it was longer. So I think it does vary between models, but also it's not a direct comparison when it's in the CSF versus iPSCs versus these aggregates in the slices. But I think it is important to think about whether a human neuron in a brain with a tangle is turning over. And I think Wash, you are going to be doing some more work on that.
Yeah, absolutely. Cool. I did said it's supposed to be the last question, but I guess Oliver would have the say. So there's a question from Marietta, Oliver, to you. And it said, how long does the stalling of capillaries persist? I'm wondering whether this develop into so-called string vessels or nanotubes, which are essentially empty basement membranes. What can you clarify that?
Yeah, I can definitely say something about the timing. I also didn't show the data. So with the multiphoto microscope, we can of course go for the short timing, but we are limited to have longer time points, or we cannot be sure that it has been flowing and restalled again. So definitely we count capillary stores if they are 5 seconds or longer. But into this earlier time points, or whether leukocytes or these neutrophils overall are a little bit slower. In those Alzheimer mice, we currently PhD students trying to address that, but we know that a large proportion of them are, let's say, from 5 seconds to 2 or 3 minutes. But some other people have also now started to look into that, and there are some that persist at least for 20 minutes. So that can be only a few, I guess, or maybe we are missing them when we image them. It's hard to say, because we probably wouldn't catch them. So I think that's really interesting regarding the string vessels. I know those the string vessels are quite common. You see them in patients with Alzheimer. So we really haven't seen vessels degenerating or becoming string vessels that have been stalled at a given time. So we don't have that many stalls where we really know it for a week or 2 or 3 or 4. So these long-term examples are we don't really have, which has to do with the long-term imaging is challenging, and then finding as the exact capillaries again is also not as easy. So we are lacking this data work. We're trying to accumulate this data to see really what is happening to those capillaries, and are capillaries that are frequently stalled, do they change to string vessels or they degenerate? We just have to see that. So I really cannot give you an answer there. So I can only say I haven't seen it yet.
Okay. Yeah, well, I guess we have to end somewhere, but this conversation can continue perhaps on Twitter, or you can email the speakers. But I have to thank all the speakers for such a wonderful presentation, and the Q&A is also fantastic. Thank you so much. And I also want to thank Tara for waiting this long. I guess I'm gonna say that Tara doesn't really need introduction, but I'm probably just going to introduce her for the sake of formalities. As you know, she's going to be giving the plenary talk of the day on synaptic changes in Alzheimer's disease. So before I introduce her, I'll probably invite her to share her screen first. Can you share your screen before we go into the introduction? Fantastic. Cool. Okay. So I know she wouldn't want me to go on and on to introduce her, but like I said, for the sake of formalities, I'll go ahead and do it anyway. So Tara is a professor of neurodegeneration and acting co-director of Center for Discovery Brain Sciences at the University of Edinburgh, and a program lead at the UK Dementia Research Institute. She obtained her Bachelor's of Science in Biochemistry and Bachelor's of Arts in French in 1999 from the University of Texas at Austin through a National Merit Scholarship. She obtained an MSc in neuroscience at the University of Oxford through a British Marshall Scholarship, which enabled her to work on environmental influences on synapse development and degeneration with Sir Colin Blackmore. After completing her PhD in 2004, Cara worked as a postdoctoral research fellow at the Massachusetts General Hospital and Harvard Medical School, where she undertook research on synaptic degeneration and Alzheimer's disease pathogenesis. And following that fellowship, she remained at Massachusetts General Hospital and Harvard Medical School as an instructor, then assistant professor from 2011 to 2013. And in 2013, Cara moved to Scotland to join the University of Edinburgh as a reader and Chancellor's Fellow. She was also awarded at that time a personal chair of neurodegeneration in 2017, actually, not at that time. Her research focuses on the mechanisms and reversibility of synapse degeneration in Alzheimer's disease, other degenerative diseases, and aging. In addition to her research, she's a passionate science communicator and regularly engages in outreach and engagement activities. She's a member of the Science Media Center, advising journalists on science reporting and commenting on new science stories. She served as a member of the Scottish Science Advisory Council from 2016 and 2019, advising the Scottish Government on science policy. And in 2018, she was elected as a term member of the European Dana Alliance of the Brain, promoting public engagement with neuroscience. And sorry, I've got 2 paragraphs else to say, so just hold on for a bit. Cara is founding member of the Federation of European Neuroscience Societies, Fenskavli Network of Excellence, Excellent Scholar. Currently, she serves as a chair of the Alzheimer's UK Grant Review Board, as well as founding editor of the Brain Communications Journal. Of course, she serves as reviewer for many journals and grant agencies. She is an advocate for increasing the rigor and reproducibility in translational neuroscience, promoting inclusivity and diversity in science, and supporting career development of neuroscientists. So I invite you all to listen to this talk and send your questions. And I would like to also say, perhaps if you have any questions related to career development as well, you could as well send it. I'm sure she'll be happy to answer them. Cara, so sorry to keep you waiting. We look forward to your talk now. Thank you.
Thank you very much, Mahmoud, for the kind introduction, and Rebecca for organizing. And a big thanks to all the speakers. I've really enjoyed the session and learned a lot, and it's been fantastic. Very hard acts to follow. So please shout if anything goes wrong with the tech, because I'm still, despite months of lockdown, not the best at Zoom, and it's hard for me to see the chat. So someone do speak up if anything goes wrong with the slides or anything. But thank you for the opportunity today to share some of our data. As Mahmoud said, I'll be talking about some of our work on synaptic changes in Alzheimer's disease. But since it's an ECR event, Rebecca said I could also sort of indulge myself and talk a little bit about career progression, with the caveat that this is an N of 1, that I'm most familiar with my own career and maybe a small case series of the people that I've helped come through the system so far. So it's not, you know, maybe the world's best career advice, but I'll at least tell you a little bit about one pathway. So first, I'll declare my interests. None of the data that I have talked about today has been influenced by any of our industrial collaborations, but it's also a good chance to thank our funders, largely the UK Dementia Research Institute, the European Research Council, and several of the research charities, Alzheimer's Society and Alzheimer's Research UK, have really done a lot for getting my lab up and going in Edinburgh over the past 7 years and keeping our work going. So what I'll talk about today is I'll briefly introduce Alzheimer's disease, although I don't really need to now. We've had several talks that have done a brilliant job introducing the disease and the importance of it. And then I'll go through a little bit of data about how we've observed amyloid beta, ApoE, clusterin, and tau all accumulating in synapses in Alzheimer's disease, both in human brain samples and in some different mouse models, and why we think that might be important, and a little bit of more recent data showing that we think glia may link some of these pathological proteins and synapse degeneration. And then finally, as I mentioned, some comments on career progression in this field of translational neuroscience. And this is a 3D reconstruction of a little bit of human brain. The red is synaptophysin staining. I agree. I think it was Henriette who said synaptophysin was a beautiful marker. I agree. In our hands, it's absolutely lovely. And this is a picture of our lab from when we could still get within 2 meters of each other. I think this was our Christmas party last year. And they're an absolutely fantastic bunch. So I want to be sure and show their picture at the beginning in case I forget or run out of time at the end. So first, really briefly, why do we study Alzheimer's disease? I think for this audience, it's probably you're already sold. But just in case you don't work on the dementias, dementia is an umbrella term for a range of symptoms. It's not a disease in of itself. It's this umbrella term for lots of different diseases that cause this core set of symptoms, which is progressive cognitive impairment over time. The most common cause of the dementia symptoms is Alzheimer's disease, then vascular dementias, Lewy body dementias, frontotemporal dementias, and there are other causes. Pretty much anything that can damage your brain in an ongoing way can cause this core set of symptoms. And one of the reasons we study it is because this is a huge global public health problem, not only in first world countries, but in low and middle income countries as well. These numbers that I'm showing here is just the nicest graphic that I've seen around in there. So they're a little bit out of date from 2015. At the minute, it's estimated that there are about 50 million people living with dementia worldwide. And as our population ages, these numbers get worse. And the cost, both to the people themselves and their families, and also the over 800 billion euros per year that we spend on caring for people with dementia, is getting worse. So we really need to find ways to develop life-changing treatments or preventions. So what goes on in the Alzheimer's disease brain? You've already heard quite a bit about this today, but there are 3 things that define Alzheimer's disease neuropathologically. And we still pretty much rely on neuropathological diagnosis for the definitive diagnosis of disease, although now there are some pretty good biomarkers that can give good hints in vivo as well. But the 3 things that define Alzheimer's disease still that Alois Alzheimer laid out in 1907 are atrophy, plaques, and tangles. So atrophy is illustrated here, a coronal section through a person who died of non-neurological conditions and a person who died with Alzheimer's disease. And you can really see the shrinkage, the atrophy, just visually. It's really evident here in the hippocampus, which is of course very important for learning and memory, and degenerates early in the disease process. And also in the neocortex, you can see that you've got drastic thinning of the neocortex and expansion of the ventricles. And this atrophy is due to loss of neurons and neuropil that we'll discuss. Microscopically, the neuropathological lesions that define Alzheimer's disease are amyloid plaques that are made of amyloid beta that accumulates extracellularly. We heard a little bit about that being produced in the iPSC neurons earlier. And intracellular neurofibrillary tangles, which is tau that we've heard a lot about from Cara, accumulates in a hyperphosphorylated state within somatodendritic compartments. So those are the 3 core pathological lesions, but they don't all appear at once all over the brain. This is a very slowly progressing disease. So this is a graph that I've sort of adapted from one of Martin Ingelson's reviews a long time ago now, that shows the amount of pathology or brain structure on the y-axis and time or aging on the x-axis. And prodromal is a phase in which people don't have any disease symptoms at all, even if you took cognitive tests and neuropsychiatric tests. MCI is mild cognitive impairment. It's a term which some people hate, but at the end of the day, it's when you can detect some cognitive symptoms, but not full-blown dementia. Not everyone with MCI is on the path to Alzheimer's disease, but about 50% of people are. And then Alzheimer's disease is when you have that full dementia diagnosis. So if we look at what's happening in the brain across this spectrum, and this is over the course of many years, you see that amyloid beta, these plaques, accumulate for, we think, at least a decade, probably 2 decades. They accumulate in the brain, and this is in the prodromal phase when there are no detectable symptoms. We think your brain can just make up for a lot of local pathology. You have a resilient brain, and you can have a pretty much full load of amyloid without any problems. But when you also have tau, the tangles spreading out from the medial temporal lobe, then you start to have these mild cognitive impairment or cognitive symptoms. So tau accumulates very slowly, mostly in very restricted parts of the brainstem and medial temporal lobe before symptom onset. But after symptom onset, you get a rapid increase in tau pathology and spread through the brain. And that's about the same time that you start to get a rapid loss of neurons in the same parts of the brain that have tau pathology. Oh, sorry, neurons are the green ones. I'm pointing to the wrong line. But what we focus on in our lab is synapse degeneration. And one of the reasons is because the synapse loss actually correlates most closely of all of the brain changes with the cognitive symptoms. And that's shown here, some of the original data from the early 1990s. Dukovsky and Sheff actually went and did some heroic counts of synapses in transmission electron micrographs from both postmortem samples and biopsy samples, and showed a tight correlation of the density of synapses with the many mental state exam, which is one of the cognitive tests used for Alzheimer's patients. And at about the same time, Terry and Mazaya and colleagues did some synaptophysin staining and quantified optical density of synaptophysin staining, and again saw a close correlation with cognitive tests, that same cognitive test. And this has been well replicated in the field. We know that in rodent neurons and in rodent brain, oligomeric A beta is actually particularly synaptotoxic. And that's when I came into the field, was about 2004, when there had been some great data from Dennis Selko and several other groups showing oligomers of A beta were particularly toxic to synapses. So the idea of what's happening in the disease is that, this is a very simplified version, obviously, is that risk factors like age, genes, and environmental risk factors somehow combine to increase amyloid beta production, or at least accumulation, either increased production or decreased clearance. But downstream of this, you get the tau pathology, and downstream of that, you get neurodegeneration and cognitive impairment. So this is what people in the field call the amyloid cascade hypothesis, and there are plenty of problems with it and questions remaining. So I'll start off here with what are the, oh, sorry, I've got a little pop-up. So one of the key questions that we're looking at in the lab is, do pathological proteins in the human brain contribute to this synapse degeneration that we think is really key for the cognitive symptoms? So before I move on to talk a lot about synapses, I want to give a little bit of introduction about what causes dementias, in particular Alzheimer's disease. And in my pretty simple mind, I lump this into three buckets, three causes or contributing factors to Alzheimer's disease. Aging is the biggest risk factor for Alzheimer's and all of the other dementias, but genes are known to contribute. We heard about presenilin 1 and APP earlier. There are rare familial forms of genetic Alzheimer's disease, but also there are risk factor genes that substantially increase your risk. And we think there are also lifestyle factors that contribute to the risk of Alzheimer's disease. And this is important because we think across large epigenetic meta-analyses that about 1/3 of dementia cases might be able to be prevented by modifiable factors, which would be massive on a public health scale. So things that are associated with increased dementia risk won't be too surprising to you. These are things like smoking, high blood pressure. We heard about just now from Oliver about the vascular factors that can influence dementia, obesity, diabetes, and physical inactivity. Social isolation, depression, and hearing loss have also been associated with increased dementia risk. Associated with reduced dementia risk are things that are generally good for you, like exercise and healthy diet, level of education, and keeping active both physically and socially. And one big caveat, since this is an audience full of scientists, it's probably not really necessary, but I always like to point out, because of the public engagement work I do talking about association, lifestyle association factors with dementias, is that studies that show these associations are good and very powerful, but they can't prove causation, right? So one of those that I like to point out here is in particular depression. Having depression is associated with the risk of increased risk of dementia later in life, but depression could either be contributing to the brain changes that cause dementia, or it could be an effect of the brain changes in the early stages of dementia. So just be cautious when you hear these associations. We need more work to really nail down the biological links between these lifestyle factors, and in fact also between aging and genes, and why people get dementia. So to briefly introduce the genetics bucket, so I just talked about lifestyle, now I'll talk about genes. Risk genes are important because we think that there's a huge genetic component to risk for Alzheimer's disease. Not only, so these are the three causative genes. They're very, very rare. Probably less than 1% of Alzheimer's cases are caused by familial mutations, although they've done a lot for us in terms of understanding the biology. But there are much more common variants that can increase your risk, albeit at a much lower level of increased risk. So instead of having 100% causative risk, some of these genes down here only increase your risk a very small amount, but they're also very common in the population. And the two genes that I'll talk about today are ApoE4 and clusterin. And these are very interesting. ApoE4 is the most important genetic risk factor for sporadic, e.g. non-familial inherited AD. And both of these genes are expressed in glia. So most ApoE4 and clusterin are most abundantly expressed in the central nervous system of the apolipoproteins. ApoE is normally made in astrocytes at high levels and secreted by astrocytes in homeostatic conditions. It's very important for lipid transport, for example. Clusterin is expressed in neurons and astrocytes, and it's upregulated in astrocytes in disease. Both of these are thought to influence A-beta aggregation and clearance, and both of these are thought to be also involved in neuroinflammation. So there's been a big move in our field lately to look at glia, and in particular microglia, because many of the genetic risk factors discovered in the GWAS studies are pointing to the idea that something changing in microglia can vastly influence your risk of dementia. But we still don't really understand the biological links between these risk gene variants and what happens in the brain to increase your risk for disease. So that's one of the things we work on actively in the lab. And just to introduce this concept of it's not all just neurons, we've talked a lot about neurons today and a little bit, of course, also about the vasculature. But because of these genetic studies, because of the epidemiology so strongly linking vascular factors to dementias, there's been a shift in the past few years, and probably across all of neuroscience. Actually, you're probably aware too that microglia seem to be taking over neuroscience. It's an exciting time to be a glial biologist, I'm sure. But when you think about Alzheimer's disease, if you think about the normal NVGU that all needs to function together for normal brain function, in Alzheimer's disease, not only do we have the plaques and tangles, but we have plaque-associated and in fact also tangle-associated accumulation of microglia and astrocytes. We have loss of oligodendrocytes and white matter and myelin damage. The synapse loss that I'll talk about a lot today may be mediated, at least in part, by microglial engulfment. We have the impaired vascular clearance of proteins, hypoperfusion, and blood-brain barrier breakdown. So there's a lot more going on than just the neurons and the synapses, even though I'm going to mostly talk about the synapses because they're my favorite. But I think it's probably one of the most active parts of the field is this question of how glia are influencing degeneration. In our particular little corner of the field, it's how glia are influencing synapse degeneration and this protein accumulation within synapses. So to talk a little bit more specifically about that, some of the work we do is inspired directly by Soyeon Hong and Beth Stevens, who showed that microglia phagocytose synapses in Alzheimer's mouse models via complement. So the postsynaptic densities in APP-PS1 mice are tagged with C1q, and then that signals the microglia via the C3 receptor to phagocytose the synapses. Normally, microglia prune synapses during development, but we think this may be reactivated and aberrantly activated in disease. And in terms of tau, there were a couple of good papers in the last year or two showing that tau pathology also may be causing synaptic phagocytosis via this complement pathway. So it's a really interesting part of the field right now. So the hypothesis that we're sort of testing that I'll talk about today is that pathological protein accumulation, particularly in synapses, drives synapse loss in a process that involves interactions between neurons and glia. So instead of the straight amyloid cascade that I introduced at the beginning, our sort of working model of what's going on in terms of synapse degeneration is a bit messier. It looks a bit like this, and I'll give you some evidence to support this messy version over the next few minutes. So how are we testing this hypothesis? As Cara mentioned, it's important to use multiple models and approaches. I think that's really important for the field, that we're ultimately aiming to do something that will benefit people with these diseases. So we're trying very hard to replicate what we see in different model systems. We use human postmortem tissue. We use animal models, traditionally mice, but we now have some fly work that I won't talk about today. It's still in very early stages. And we use cultured cells, both human iPSC and mouse and human primary cells. So as I go through the experiments, I hope I remember to stick a little icon up in the corner about which model system we're using, because I might jump back and forth and be a bit confusing. One of the methods that we use as our workhorse in the lab to look at synaptic protein accumulation is the array tomography technique. This was developed in Stanford by Steve Smith and Christina Makeva several years ago now. And then we adapted it for use in Alzheimer's mice and human postmortem tissue. And the actual details are quite tricky, but the crux of it is you embed the tissue in a hard plastic resin, so we can section it in ultra thin sections, and that overcomes the axial limit of light microscopy, because synapses are too small to accurately look at colocalization without this. And what we observed with this is that A beta accumulates in synapses in human brain, and it's associated with synapse loss near plaques. This is work that I started in Boston with one of my first MD PhD students, Robert Kofi. And what Robert and I did was use the human array tomography, human brain, embedded it for array tomography, sectioned it, stained for presynapses in red here, postsynapses are green, and A beta in blue. And Robert went and measured all the synapse degeneration. And we saw that as you got closer to a plaque, you have a loss in synapse density compared to this average for the control cases. And further, why I wanted to show this data, even though it was published several years ago, is that we saw that ApoE4 carriers had worse synapse degeneration than ApoE3 carriers. And when we went and looked a little bit more closely, we also saw that the synapses that contain both ApoE and A beta are much higher in the ApoE4 carriers than ApoE3 carriers. So one of the things we think ApoE may be doing is driving toxic oligomeric A beta to synapses in the human brain. More recently, Rosie Jackson, when she was a PhD student with me in Edinburgh, before she actually went the other way to MGH, she's now a postdoc there, Rosie looked at clusterin. So clusterin is one of the other risk factors that I talked about. She stained clusterin, presynaptic terminals, and A beta, and found that very similar to ApoE, there is an increase in presynaptic terminals with clusterin, and this occurs more near plaques, and it occurs more in people with an ApoE4 allele. One of my favorite parts about this whole study, I'm not showing you here, was that we replicated the data that Robert saw about synapse loss near plaques and an ApoE4 effect on this in humans in a whole different cohort of people in Edinburgh. So I really liked to see that we could see the same effect again. I don't know why that makes me so happy, but it really does. Replication within our own lab makes me much happier than it probably should. So this is linking now another risk factor to accumulation of amyloid beta in synapses. Recent work with Caitlin Davies, we've been looking at synaptic tau accumulation, and we've done this a few times. We worked with Patrick Verstraeken, and we worked when Ellie Pickett was a PhD student in the lab to observe tau in synapses, but Caitlin has taken this a step further and looked at a whole lot of cases, looked at the effect of ApoE, looked at both pre and postsynaptic terminals, and she's also become an expert at MRIS rendering, which also makes me very happy. You can see these beautiful presynaptic terminals, postsynaptic terminals, and here's one presynapse that contains tau. These little snowmen, what we call them in the lab, you can see the apposition of postsynaptic terminals quite nicely with the array tomography. And what Caitlin has found recently, when she looked at paired synapses, and by paired I mean this is presynaptic terminals that had a postsynaptic density within 0.5 micron. So without electron microscopy, we can't say for sure that they are a synaptic pair, but we can say that they're close enough. So that's just another filter to try and rule out random noise. We do have several different filters to try and remove noise from our images. And when we look at the percentage of presynapses that have ALT50, which is a misfolded tau epitope, what Caitlin observes is that there's an increase in AD compared to controls. But here, look at the controls without an ApoE4 compared to the controls with an E4. There's an increase in synaptic tau with E4 even in controls. So there's this sort of stepwise increase in presynaptic tau as you go from control without an E4 to control with E4, AD without E4, and AD with E4. And that pattern is repeated when we look at the percentage of postsynapses that contain tau as well. We also have had a look at 2 brain regions using unbiased proteomics, because we had sort of really targeted experiments in the past looking at tau and A beta, but we wanted to get a better idea of what else might be happening in human synapses. So Rosie Jackson and Raphael Hesse homogenized these 2 brain regions, prepared synaptoneurosomes, which is actually a pretty simple method of filtration after homogenization with a couple of spins, and we get these little pairs of presynapses and postsynapses that sort of seal off. And then we sent those off to Dundee, studied them all to do mass spec, and looked at what happened to the proteins in human synapses. And a lot happened. We identified over 5,500 proteins. But what I'll highlight today is that there's this increase in pathways involved in neuroinflammation. So when we just looked at an unbiased way with the bioinformatics, some of the top pathways that changed were CD40 signaling, IL-6 signaling, and acute phase response signaling, which are all involved in neuroinflammation. And you can see these are all most upregulated. Up is red and down is green in these images. Most upregulated in the temporal cortex of ApoE4 carriers when you compare AD to controls. And we did a little post-hoc analysis of complement-related proteins, and most of them that we observed were up in AD compared to controls. So this was one of the reasons that we sort of started looking at the glial involvement in these processes. And another reason is a fantastic PhD student, Makis Sioras, he's in love with microglia and really wanted to study the role of microglia in synapse loss in AD. So Barry McCall and I co-supervise Makis because Barry is an actual microglial expert and I look at the synapses. And Makis was able to stain microglia, these are CD68 positive microglial lysosomes, presynaptic proteins, and the blue is happy, and it also labels plaques because we have some thios in there. And what Makis observes that he can find co-localization of synaptic proteins within lysosomes of microglia and this occurs more often in AD and more often in the temporal cortex. So this is some evidence from human postmortem brain that microglia might be ingesting synapses. And this is directly in line with what Soyan and Beth saw in mice, but hadn't been shown yet in humans. This co-localization of synaptic proteins within microglia increased near plaques. And I won't go into the details of this, but this is just some evidence that A-beta, because this is around plaques, might be causing synapse degeneration in cooperation with microglia in this case. And Barry was able to work with Mike Daniels. I'm just seeing if this movie's going to work. Yes, with Mike Daniels and Barry McCall. Oh no, the movie doesn't work, sorry. If it worked, you would see red coming up. So Makis was able to culture microglia. Laser pointer back. And when he cultured primary mouse microglia and then fed them synapses isolated from either control or AD cases, what he found is that primary mouse microglia eat human AD synapses more and faster than control synapses. And that's shown by the area under the curve and the time to half max. We also got really lucky, and in collaboration with Veronica Moran and with a couple of the neurosurgeons here in Edinburgh, people who were undergoing surgery for tumor or epilepsy removal donated the bits of brain that would have normally been thrown away that were normal appearing brain. And Makis and Veronica were able to culture microglia. So these are primary human adult microglia now. And we saw a very similar thing to what we saw in the mice, that they eat synapses more and faster than controls. And of course, this is rare. So this is the data from one person, but we've now seen this again from another donor. So this is some evidence that it's not just an artifact of mouse microglia. And then more recently, like I mentioned, Caitlin's looking at tau in synapses, and Caitlin was interested in the ApoE effect, and since ApoE is expressed heavily in astrocytes, she went and stained in human postmortem tissue astrocytes, synapses, and tau. And she can find in purple or magenta astrocytes here, she can see presynaptic protein and phospho-tau. And when she quantified this by burden, what she found was kind of similar to what she saw with the tau accumulation in synapses, that the GFAP positive astrocytes that contain synaptophysin is increased in ApoE4 control carriers and is increased in AD compared to control. We also normalized this to GFAP burden to see if this is just an artifact of there being more astrocytes, and there is still an effect of both AD and ApoE. And when Caitlin looked at GFAP and tau co-localization, so this is tau inside the astrocytes, she saw a little bit of an effect of plaques, an effect of AD, and an effect of ApoE4. So she also looked at synapsin and tau, so co-localized synapses and tau protein, and again saw an effect of plaque, AD, and ApoE4 on this glial or astrocytic, probably engulfment of tau and synapses. So we've also looked in a mouse model to look at whether we can explore this interaction of glia and synapses and protein accumulation in synapses. And I won't show all of these data, but Ellie Pickett, we published this last year in Cell Reports. We developed a mouse model that has both plaques and human tau, and we can turn the human tau off. And they developed this phenotype of hyperactivity over time as they age. And what we found is that with RNA-seq in these mice, we saw that the mice that have both plaques and human tau have an increase, like we saw in the human synapses, of pathways involved in neuroinflammation. And this is just giving some evidence that with A-beta and tau, there are some neuron glia interactions that might be involved in synaptic phenotypes. This neuroinflammatory phenotype was really driven by the APP-PS1 transgene, by the plaques, because it wasn't present in the tau-only mice, but it was present in the APP-only mice. And when we turn the tau off, we could recover the behavioral phenotype, And we also completely reversed this transcriptional phenotype. So all of these things that were orange, which was up-regulated of neuroinflammation in the mice with both plaques and tau, are back to normal or below normal when we turn the tau off. So we think that there's a complex interaction between A beta, tau, glia, and synapses in these, at least in the mice, and we have some support for this in the postmortem human tissue as well. So to summarize the scientific parts of the talk, I've shown you in sort of a whirlwind tour that A beta, tau, ApoE, and clusterin all can accumulate in synapses, where they're associated with degeneration of synapses. Our proteomic study showed increased levels of proteins involved in neuron glia signaling and neuroinflammation. We've shown that microglia phagocytose human AD synapses more than control synapses, and we've shown that in both human postmortem tissue, adult primary microglia from humans and mice. And also, I haven't shown the data today, but we have some data from collaborators in Cambridge showing a similar phenotype with iPSCs from humans. And finally, I've shown you that astrocytes contain some synaptic proteins in human brain and contain some tau. So altogether, I hope I've given you some hints about why I've made this little bit more complicated version of the amyloid cascade that puts neuron glia interactions somewhere in between A beta and tau in causing synapse degeneration. So just to finish with a few minutes of talking about career development, I wanted to show you a timeline. So there are ups and downs in everyone's career and everyone's productivity, but I'll start with just, oh, hello, here's my dog, sorry. Here's where my sort of career timeline, as Mahmoud already summarized. I got my PhD in 2004, I got my undergraduate degrees in 1999, and this all looks really nice and linear, right? So the way he laid it out, it sounds like a perfect linear progression. But then if you look at productivity in terms of papers per year and grant income per year, you can see that this fluctuates quite a lot. But generally, I'm glad to say it's gone up a bit during my career from no papers when I started to some papers every year now. But then if you overlay what's happening in life, you can see that the paper dips coincided quite nicely with having my first kid and my second kid. But then look at this is moving across the Atlantic from Boston to Edinburgh. There was a huge starting over productivity dip there. So this is just to say that there's going to be, of course, ups and downs in every career. And that leads into my next point about career development is resilience, really, because I love being a scientist. I absolutely love being an academic researcher, but there's a lot of failure and a lot of rejection. So you need a lot of resilience to succeed or to survive in this career or to enjoy it. So I would say about resilience that failure and rejection is sort of part of the process, and failure is also part of how we learn. If it was easy, we wouldn't have to do such complicated and expensive experiments. So we are going to, by definition, fail a lot of the time in our lab work. And I would also say rejections always hurt, but I think ultimately, usually rejections do help improve the science as we move along. My final thought about resilience is please ask for help. Ask for support when you need it. And my example of this is one of my very first papers in the Journal of Neuroscience. I was in a physiology department in Oxford, and we had an idea to check for BDNF levels in our Huntington's disease mice. And my mentor, Tony, said, do Western blots. So I basically reinvented the Western blot. I learned how to pour my own gels from scratch and make acrylamide gels and combs. And this was probably the stupidest thing I could have done as a PhD student, because there was a biochemistry department across the street. They probably could have saved me a year of time by walking across the street and asking for help running Western blots. So you need resilience, but you also need to ask for help when you need it. Another thing that I think is important for career development is engagement. Engagement both with other scientists, but also engagement with the wider world. So we usually engage typically in networks and conferences like we're doing now. I think it's important that we promote rigorous and reproducible science, and we engage with our publishing partners, with our grant review boards that we work with to try and encourage this, because it really is important for neuroscience to progress. Of course, what we're doing today, I think it's really important for all of us to encourage early career neuroscientists. There's never too early to be a mentor to other people who are coming through. And it's important to explain to the public that the ultimately fund most of what we do, and also their continued goodwill in a pressure policy is to continue funding what we do. And on that point, I think it's also very important that we engage with government. I mean, we've all seen what's going on in the world now with politics, and it's a difficult time. And I think it's really important that we try and make sure that expert is not a bad word, that we're doing our best as academic scientists to make the world a better place. So I've just shown some examples of the kinds of engagement that I like and think are important. Mahmoud mentioned I'm a member of the Fence Cabling Network, which is a brilliant early career to mid-career network of scientists across Europe. I'm trying to push rigor and transparency and reproducibility through the work that I do with grant review boards and with the journals that I work with. I try a little bit to work with policy and advise best I can based on our expertise in our lab, to try and influence the future of dementia research. And also I try and engage a bit with the wider community, which at first is very nerve-wracking, but I think it's also very important, and it's sort of easy. Once you get into it, it's a lot less work than preparing a scientific talk to talk about in general terms of dementia research. Once you've prepared it once, it becomes quite easy. So my sort of final couple of thoughts, I hope I'm not running over yet, are I think it's really important that we think about inclusion. Diversity of thought is important. We should, as neuroscientists, really know and appreciate this. So I think the more diverse people and groups and backgrounds that we have, it's going to only help us in terms of our discovery and our outputs. And of course, it's also one of the best things about being a scientist is we get to meet tons of interesting people from all over the world, including, here's some more pictures of our group. We are fun group of people to work with, and these are just a few. Like our typical neuroscience talks will include historical greats, which are usually, historically, because of the way the world was, older white men. But now if you look even a tiny bit, you can see examples of heroes, of people doing amazing work who are from all different backgrounds. So these are just a few of my personal heroes in neuroscience who aren't quite from the normal mold of the old school founding fathers of neuroscience. And of course, it's important to enjoy life outside of science. I know that should go without saying, but some of us get so invested in what we do that we forget that there's a wonderful world out there, and that we are not just a sum of our papers and our experiments, but we're whole people. So I hope that this half hour or so has convinced you that our group is great. We've used lots of approaches to look at how pathological proteins damage synapses in Alzheimer's disease, and I'm really excited about the work that we're doing. I hope I've also convinced you that being a scientist is a great calling, but it does require resilience, and you are going to get rejected throughout your entire career if you stick with an academic path. And finally, I hope you'll go away with the idea that future breakthroughs and making a real difference with our neuroscience needs collaboration between scientists and with the wider world. So with that, I will say thank you again. A lot of the work I presented is based on these two great PhD students, Marcus and Caitlin. And here's the whole group. And of course, we have collaborators all over the world who've been absolutely instrumental. And a massive thanks to our patient donors and their families who make all of this work possible. So thank you very much.
Fantastic. Thank you very much, Sarah. It's always great listening to your talk. While we wait for questions to come in, I'd like to ask one scientific question and a career question. So for the scientific question, I was wondering, in your data, you showed that synaptic degeneration is somehow associated with the accumulation of these aggregates at the synapse. I was wondering whether non-aggregate accumulated, without accumulating at synapse, some sort of processes. So for example, maybe oxidative stress or something. What contribution do you think those processes, those mechanisms do on synaptic degeneration? Do they have to always have like aggregates at the synapse for the synapse to degenerate, or is there something happening?
You may have to repeat part of that. My dog lost his marbles about halfway through the question. I don't think it's just synaptic aggregates, but I kind of missed a bit of the question, sorry.
Yeah, so basically I was wondering, because you've shown that when you've got these aggregates at the synapse, it's associated with synaptic degeneration. So I was wondering what contribution other processes have for that synaptic degeneration that have nothing to do with those aggregates accumulating at the synapse?
Yeah, that's a great question, because I get really focused on these aggregates because they're so fun to look at, but you're absolutely right. I think there are going to be a lot of processes that are involved in synapse loss and degeneration that have nothing to do with local accumulation of A beta and tau. for example, in ALS, there's a pretty well-established idea of dying back. And I think that probably applies to all of these neurodegenerative diseases. If one neuron dies, all of its connections are lost to the downstream neurons. And we know that the inputs to cells have an impact on their function. So I think just changing the network in any way, and that could start with synaptic protein aggregates, but it equally could very well start with changes in cerebral blood flow, changes in the way microglia or astrocytes are supporting the network, or any other sort of damage to the brain. Any change in the network will, by definition, impact, I think, on the synapses in the connected neurons. So I think there's probably a lot going on. We've only scratched the surface. Thank you.
Absolutely. And the career question. So if you look at your career, you see that you're quite successful compared to a lot of people. And I was wondering whether there is a kind of code to use to, while you're passionate, but at the same time doing lots of other things to succeed in doing science. Because looking at what you do, you could see that you do a lot of different things, but yet you are focused and you are achieving what you're doing.
Yeah, so it's the question about balance of focusing on your science versus the extra things.
Yeah, there's any formula or whatever.
Yeah, I wish there was a formula, but I don't think there is. I think everyone is different, and it takes a lot of trial and error to know what's going to be both enjoyable and useful and good for your career, right? So some people focus really entirely on their science and do a brilliant job and become superstars in the field and make a big difference. And I think that is one incredibly valid path. Other people focus on teaching and mentoring. Other people focus on engagement. And I tend to just throw things at the wall and see what sticks and see what I find rewarding and hopefully not damaging to my career. And it's been a real learning process. I found out along the way some things that I definitely don't like and will not pursue. I mean, so one example of that is I don't enjoy internal leadership and management within the university. I never want to be the head of our university. It's important and it's a necessary job and people do a great job mentoring. I don't find it rewarding. I find it very like everybody's just mad at you all the time. So I've tried a few things because I've been asked, and then I've decided to step back because it's not for me. So the things that I find quite rewarding, so I think in that way it's individual, right? Things that work for you and that are important and meaningful for you. And I find the public engagement quite fun and low. You get a lot out for not putting much in terms of time. So I like that one personally because I think it's important to engage with society, and it also, once I have a system for doing this, it's a very like a half hour a week, and then it's done.
Absolutely, thank you very much for that. So I would encourage everyone that has questions to drop their questions. This is a great opportunity to get them answered. So we've got a couple of questions. There's this question that asks, what do you suggest to someone who wants to get started with science communication with the public? Blogs, social media.
Yeah, that's a great question. So to get started, media training is not a bad idea. It's something I wish I had done before I was sort of sitting in front of a camera, because it's not so much about I think that media training is useful because you learn how to prepare in advance. So my tip for preparing in advance, if I'm going to be on the radio or something, is to prepare 3 points of things that I want to say. And also the media training can give you an introduction to people who are involved in this type of thing. One good intro to people who want to get involved in public communication of science is to volunteer for things like science festivals. Around here in Edinburgh, there's always people looking for volunteers for public engagement. So getting in volunteering with public engagement is a way of seeing whether you like it, getting a little bit of experience on your CV. And the sort of the way I got into the Science Media Centre is they sort of approach experts in certain fields. I don't know where they get their names from, honestly. I mean, if they come with a question of that I don't know the answer to, I often say, well, my colleague would be great to tell you about this. So it's probably like that. They build their network of experts that way. So sometimes you get asked, but another really good way to get into public engagement is with your own work. If you publish a paper, send it to your press office and say, look, this is a paper. I'm a big believer that we should be publicizing what we do, even if it's very basic neuroscience, because it's very important. And it's important that people understand that the fundamental science is important. So I tell a press office every time we write a paper. And sometimes they don't write a press release and sometimes they do. But if they do, it's a great chance because you'll get contacted. And then you can talk about your paper in whatever, local radio or blogs, things like that.
Cool. So we've got two questions that are science related. And I think in terms of time, we're probably running a bit short. But so one of these questions that was the microglial phagocytosis of synapse in human samples region specific?
No. So there was a little bit of a difference qualitatively. You could see there was more phagocytosis in the temporal compared to the visual cortex, which is what we expected because the visual cortex is less affected and affected much later. But from memory, when we did the linear mixed effects modeling, I don't think there was a region effect, a significant region effect. It looked like there was more, but I don't think it was significant. Short answer, sorry.
So we've got this question. I'm wondering what your thoughts are. What are your thoughts on targeting on that aging itself may be a driving force of neurodegeneration, and that we will need to target the aging process itself to develop causative rather than symptomatic treatments? I'm not sure whether I get it.
Yeah, that's another fantastic question. That's aging. It for many years was like the elephant in the room in dementia research because everybody knows age is the biggest risk factor and nobody knows why. So we've been working a lot on that actually, and it's really interesting stuff. So we're using the Lothian birth cohort, which is an aging cohort study here in Edinburgh, people who took an intelligence test at age 11, and then our colleagues in psychology, Ian Deary and his team, have followed them up through their 70s, and a bunch of them agreed to donate their brains post-mortem. So we're looking at them now to look at people who had really healthy cognition when they aged, people who had age-related cognitive decline, and then of course the people who have frank dementia. And we're trying to understand what happens compared to younger people in the brain as you age. And the idea behind this, which has been kind of fun, is we've kind of turned the question on its head. Instead of degeneration, we're looking for resilience factors, because maybe if we could boost resilience factors and find out why some people's brains can cope with age better and cope with pathology better, maybe we could find ways of prevention. I think prevention would be so much better than symptomatic treatments if we can crack it, if we can figure it out and find something safe enough to give to everybody before we get to 65, 70. It'd be great. It's a good question.
Absolutely, absolutely. I'll probably ask the last question before we round up this. I was wondering, I think you recently authored a paper about the developments in the field in the last 2 decades, something like that, together with other colleagues. I was wondering what your thoughts are about the field going forward. What areas do you think would be exciting, or what are your thoughts about the field generally?
Yeah, that's a good question. So I think in terms of our particular corner of the field in dementia research, right now the big wave is understanding this non-neuronal contributors to neurodegeneration. I think so. That was non-existent really when I first started in the AD field. We all knew that there was gliosis, but we assumed it was an effect and not a cause. And there's been this massive change in mindset, and now we're chasing what's going on, integrating the whole NVGU and what's happening in dementia. But more broadly, it's so hard to predict, right? I mean, optogenetics and CRISPR sort of changed the game for all of neuroscience, and I really don't know what's going to change it for the next 20 odd years or in the future. But that's why being a scientist is so fun, I think. It's just you get to be right there and see things as they progress.
Absolutely, absolutely. Okay, so I think we haven't got any more questions. I would like to really thank you, but it shouldn't be left for me to do this thanking. I'll have to invite Rebecca back to end this. But for me, I would like to thank you for giving this fantastic talk and the career advice. They're always lovely. And to every speaker that we've had in the ECR session, this has been a great meeting. I really enjoyed it, especially with COVID. Not being able to go to different meetings, this really helped because it's really focused on neurodegeneration, and we've had so many different talks that gives one more inspiration about what you can do, and also the networks of people that you can contact when you want to talk about your science or your career or something. So thank you very much. Because already I've been chatting with Cara for some time about some things that I'm doing. So I think this kind of opportunity creates this communication opportunity between not just leaders, but ECRs as well. So thank you to Potentech as well as Rebecca for organizing this, but now it's up to you to end it. Thank you.
All right, thank you so much, everyone. Really great talks and really great engagement for the audience as well. And Mahmoud, you're an absolute star of a host, so thank you so much. It's been a real success, and yeah, I just really hope everyone connects on Twitter, keep the collaboration, keep the communication, and maybe one day we can all meet for real at a actual physical version of this. But yeah, I'll end it now. But yeah, thank you so much, everyone. And it was really nice to meet you. Bye.
Bye, thank you.
Bye.